In this topic, we're going to discuss the evidence for semi-conservative replication. So we're going to discuss the possible mechanisms of replication, the Griffith's experiment, the Hershey-Chase experiment, and the Meselson and Stahl experiment. Now, in the 1950s, no one knew exactly how DNA replicated. Three possibilities were suggested. These include the conservative, the semi-conservative, and the dispersive models. The first is the conservative model. This suggested that the parental DNA remained intact and that a separate daughter DNA copy was built up from new molecules of deoxyribose, phosphate, and organic bases. Of the two molecules present, one would be of entirely new material, whilst the other would be entirely original material. Then there's the semi-conservative model. This suggests that the DNA molecules split into two separate strands, and each of them would be copied to form a mirror image. So each of the two new strands would therefore have one of the new material and one of the original material. And then finally the dispersive model, which predicted that parental DNA would be broken down and the nucleotides replicated before being dispersed randomly throughout the new molecules. So the new molecules would contain both new and original material, but this would be randomly distributed and not necessarily with equal amounts of old and new material in each molecule. So if you look at the three possible explanations, you'll notice that the distribution of the old DNA is different in each model. So to find out which mechanism is correct, you need to label the old DNA and in some way look at how it's distributed after replication. So two scientists managed to achieve this, Meselson and Stahl. Now I mentioned that Meselson and Stahl's discovery was important, but there are other scientists who also did experiments that are important. These include Griffith in 1928, Hershey and Chase in 1952, and then finally we're going to look at Meselson and Stahl in 1958. So Griffiths did one of the first experiments suggesting that bacteria are capable of transferring genetic information through a process called transformation. So he showed a transforming principle which causes a change in the organism. He used pneumococcus strains, and there was a rough strain and a smooth strain. He found that when he injected the rough strain into mice, they lived, and when he injected the smooth strain into mice, they died. So in his experiment, when he injected the mice with the smooth strain, the mice died. But when he injected the mice with the rough strain, the mice lived. Then he mixed the dead smooth strain with the live rough bacteria, and he found that the mice died. So from this experiment, he concluded that some part of the smooth bacteria had been taken into the rough living ones and made them pathogenic. So today we know that this is DNA. Let's have a look at Hershey and Chase, who did an experiment in 1952. So they gave evidence towards the hereditary material being DNA, not proteins. And they used viruses called bacteriophages that injected their DNA into bacteria. So they used these bacteriophages grown in radioactive phosphorus and radioactive sulfur. So the radioactive phosphorus is only found in the DNA, and the radioactive sulfur is only found in the protein. And you know that a virus has got the DNA inside and a protein coat on the outside. So what do you think will happen if only DNA is injected into the bacteria? Which radioactive element will you find in the bacteria? Only radioactive phosphorus will be found in the bacteria. So let's have a look at what the results are. The bacteria were infected with the virus, and the genetic material was inserted into the bacteria. So all virus coats were removed using a centrifuge, leaving only the infected bacteria. So bacteria contained radioactive phosphorus, not sulfur. This means that no protein was passed into the bacteria and no sulfur was passed on to the next generation, radioactive sulfur. Right, let's have a look at Meselson and Stahl. They studied the different methods of replication. Remember that they wanted to label the old DNA somehow to see if it replicated by conservative, semi-conservative, or dispersive mechanisms. So they did this by using an isotope of nitrogen, which we call heavy nitrogen, nitrogen 15. E. coli was also used, so let's have a look at what they did. Cells of the bacterium E. coli were grown on a medium containing a nitrogen source, made up of the common form of nitrogen, nitrogen 14. This sample acted as the control, as you can see on the left there. Then some bacterial cells were transferred to a medium where the nitrogen was of the heavier isotope, nitrogen 15. So they were grown on a medium of ammonium chloride that contained, the ammonium chloride had nitrogen 15 in it. So the bacteria were grown, during which time the nitrogen that they needed to make new DNA came from the growth medium, and hence the N15 type. The resultant DNA was therefore heavier than that formed by the bacteria grown on the medium with N14. So if you look at this diagram here, the bacteria's DNA on the right would be heavier compared to the one on the left. So this was the second control. Now, after many generations, the DNA of the cells was almost exclusively of the heavy type. We're looking at the right. So what did they do next? Well, the samples of the heavy DNA bacteria were then transferred to a medium in which the nitrogen source was N14. The bacteria were grown just long enough for the cells to divide once. A sample was then removed, and the DNA of the bacterial cells was extracted and placed in a solution of cesium chloride and centrifuged. Now, because cesium chloride is heavy, it sinks towards the bottom of the tube during centrifugation, and a density gradient is established in the tube. The DNA molecules will sink in the gradient until they reach a level where their density equals that of the cesium chloride. So an extract of DNA was taken after 2 generations and finally 3 generations and treated in the same way. So the DNA molecules sank in the gradient. Now the DNA containing the N15 is going to be denser, so it's going to sink to the lower level in the tube compared to N14. Now, after 1 generation, the bacteria produced DNA that was midway between the light and the heavy DNA. So what do you think this meant? This meant that one strand of each molecule of DNA contained N14 and the other strand contained N15. Now, after 2 generations, 2 bands were formed. One with the density of light DNA and one with an intermediate density. So this indicated that it contained only N14 DNA for the light density strand, and half N14 and N15 for the intermediate density band. Then after 3 generations, there were 3 times as much DNA in the light band than in the intermediate band. So let's have a look at what this means diagrammatically. So you can see that at generation 0, the resulting DNA will be heavy DNA in both the strands of the double helix. Now in the first generation, the DNA will comprise 1 heavy and 1 light strand, giving a hybrid molecule. In generation 2, there'll be 1 light and 1 hybrid DNA molecule. In the third generation, you can see that there are 3 times as many light molecules as there are hybrid molecules. Here's another way of looking at the data. You can see that in generation 0, both strands are heavy. What about in the first generation? Can you see that the DNA is less dense than generation 0? So what does this indicate? It means that the DNA comprises 1 heavy strand and 1 light strand, so it's a hybrid molecule. Now, can you interpret the data for generation 2? It means that there's 1 light molecule and 1 hybrid molecule. What about generation 3? There'll be 3 light DNA molecules and 1 hybrid molecule. And finally, generation 4, you'll find that there's 88% of the DNA will be light molecules, whilst only 12% will be hybrid molecules. So this gives evidence to semi-conservative replication. So in summary, we've looked at the different experiments. So if you remember, Griffith did the experiment with pneumococcus on mice. He found that there was a transforming principle that is taken up and causes a change in an organism. Hershey and Chase did an experiment using bacteriophages and found that only DNA was injected, not protein. So DNA must be the hereditary material, not protein. And then finally, Meselson and Stahl proved that DNA replication was semi-conservative. So they used nitrogen 14 and nitrogen 15. So last slide. Here's a lovely diagram of the Meselson-Stahl experiment. You can see that DNA is replicated semi-conservatively by where the DNA strands contain heavy and light nitrogens. And that concludes our lesson. The end.